Please feel free to ask questions at any point, interrupt. Necessarily can make this very formal. First of all, I want to give you an escape clause. If you're here to gain wisdom about Darwin, you're in the wrong lecture. I take Darwin's evolution not as a theory, but simply as a statement of truth. It's the way biology works. And simply a description of the whole process of how life, essentially, the one thing that it's successful at is replicating itself, and what it gets rewarded for is having progeny, and that those progeny have progeny. And therefore, we see the continuation of that species and interaction of that species with the environment so as to then make yourself fittest for that particular environment at that particular time. So in our laboratory, we take it simply for granted. Everything we do, we know has to come through evolution, and that it's the process. And often we're amazed, essentially, at the spectacular things that have occurred over time to give rise to us being able to converse and think about things. So instead of talking about Darwin, what I'm simply going to talk about is what we do in the lab and give you a feeling for the kind of questions we do, and then you can see, and I'll let you introduce Darwin to that work and see how essentially we take cognizance of evolution in everything we do and everything we think about. So, what I want to do is simply give you two stories of ongoing research to give you a feeling for what we are trying to, the kind of questions we kind of address, how we address them, and the kind of tools we utilize to address them. And I'll try and explain in such a way that anybody can understand it. Mainly, all of what we do is very, very simple. It's simply often cloaked in jargon, and therefore seemingly complex, but in actuality, the thought processes of what the kind of questions we're addressing are very straightforward, and I'll try to explain them in such a way that they are understandable. Now, we do a bunch of stuff in our lab. We work on different aspects of biology and medicine. And I'll give you, one is, first of all, our patients are always mice. And that's one of the things that's where you can see that Darwin enters in. In terms of gene content, mice and humans are 99.9% the same. So whatever we find out in the mouse is actually usually directly applicable to humans. And so that allows us essentially to model, for example, diseases in mouse in much greater detail than is ever possible in humans. And then we can use that information to understand the disease and then as a platform to develop therapies. So that's one aspect of what we do in the lab, and I'll give you two examples of that. We're going to look at a mouse model for cancer and a mouse model for a neuropsychiatric disorder. And then the other things we do in our lab is simply technology development. If you look at how science progresses, it's not a curve, but it's actually a series of jumps. And those jumps are always associated with new technologies coming in that you did not anticipate, and thereby allow you to see things that you previously were not able to see, and measure things that you previously couldn't measure. And so one of the aspects of science is always to try to increase the level of technology so that now we can have a broader spectrum of questions we can address that were not addressable, say, only a few years ago. And so that's another aspect of what we do in the lab continually. So in terms of modeling cancers, the first thing you want to ask yourself is, what are the parameters that we want to make the model be applicable to. And the first thing about cancers is, when does it start? We're not born with cancers, and so we have to model it so that you're not born with a cancer. You have to pick a time when that cancer starts. And depending on the kind of cancer, some cancers affect children, some affect old age, and depending on the kind of cancers, then you want to model it appropriately. The time of induction, I've just mentioned that different cancers arise at different times. So the time of induction is important, and mostly we're not born with it. And what that means to us is that it should be modeled as a somatic disease, which means it's not in our genes, it's not in every one of our cells, but it's in the cells that make up our body. That's the soma. Stoichiometry is important. That is, how many events are occurring and how many copies of that particular insult is present in a cell. In cancer, normally you have multiple events having to occur. It's not that a single event occurs and then that gives you cancer. You usually have to accumulate in a given cell 4, 5, 6, 7, 8 events. And that's why most cancers fortunately take a very long time to develop. Because it has to have multiple things happen to that cell in order to have that insult happening. And then finally, the most important is point 4, and that is the cancer specific to a particular tissue. That is, you have breast cancer, or skin cancer, or liver cancer. It's not in every cell of your body, it's in a specific place in your body. And further, the interaction of the cancer cell with that tissue in that particular spot is important to the birth and continuation of that cancer. Okay. So what we've chosen is sarcomas. And one thing you might ask, why sarcomas? Sarcomas are not as common as carcinomas. Carcinomas is what most humans get. And carcinomas, again, fortunately, takes a long time to develop. But sarcomas are extremely aggressive, and they affect primarily children and young adults. And because they're not common, drug companies aren't as interested in them, and therefore they did not get as much attention, essentially, as carcinomas do. So that's why we are modeling carcinomas, and these are the reasons. It affects the clinical population, I've already said, children and young adults. They're extremely aggressive. They're not extensively studied, but we do know how the cancer starts. And in most cancers, we don't have that luxury. If you're a modeler, you like to start at the beginning, rather than somewhere in the middle or at the end. And so in this case, we're very fortunate that in many sarcomas, not all, but now there are over 25 different sarcomas that have been identified, and each has a common way of starting. And it's by 2 chromosomes breaking and then rejoining with opposite partners. And so what we call that is a translocation. So a chromosome is simply a collection of genes. Imagine as pearls on a string, and then you break them and then you join them together. But if you have several different strings, what you're doing is breaking one, breaking the other, and then joining them with opposite partners rather than the same partner again. Okay? So everything's okay when you do that, except where you actually break and join. Because often you'll break in the middle of one gene, you'll break in the middle of another gene, and now you join them together and you make a hybrid gene that never existed before. Okay? And it's that product, essentially, that's responsible for initiating sarcomas. And we call them a translocation. So that's one big advantage, is that we know what the starting event is. And finally, they're not as complex as carcinomas. One thing that happens in carcinomas commonly